Welcome back, Year 12s, to a video about biochemical pathways and photosynthesis. Alright, we'll just do a quick recap of enzymes for those who either haven't done it or have forgotten about the basics of enzymes, because they do act on biochemical pathways, as you can see in number 8 here. Alright, so let's go through it quickly. So number 1, they're reusable. Alright, they do not get depleted once they're used. The only time they can get deformed and depleted is if they get denatured. Alright, number 2, specific. Most enzymes will only bind to one specific substrate. Okay. 3, enzymes are reversible, so there's generally enzymes that will break down some sort of substrate, so one thing into two, or there's going to be enzymes which, of course, put two substrates into one product. Okay, and those different reactions are going to be called anabolic if they're building a molecule with, of course, some sort of substrates, or a catabolic reaction if they're breaking down a molecule, all right, into substrates. Cool. Number 4, speed up, not create. They're catalysts, all right, so they're speeding up the rate of reaction. If you remember, a burger lasts for 100 years, unless, of course, you have enzymes, then it digests in 24 hours. All right, they have an active site, number 5 here, so a ridge in which the substrate fits into, and then it comes out of when it's, become a product. 6, they are proteins. Most enzymes are proteins, okay? So we just think of them as proteins. 7, they are all catalysts, as you can see, but some catalysts are not enzymes, such as, cofactors and coenzymes and other things. All right, they act on an entire biochemical pathway. That's why we're looking at it in photosynthesis right now. All right, most of them end in A's. All right, the ones founded in the last 30 years or so, well, 40 years actually. Previously, like pepsin, all right, they don't end in A's, but most of them do, like catalase, polymerase, ligase, lactase. And of course, in a chemical equation, they're going to be above the arrow. So an example of that would be light in photosynthesis reaction. So biochemical pathways of C3 photosynthesis. Cool. So we've got a bunch of different dot points now. All right. So if we look at plant cells first, plant cells are thought to be as green, but realistically they're this transparent white-ish sort of color, like sticky tape. All right, which is also where cellotape's made from, cellulose, cellotape. Anyway, so really the part that's green is your chloroplasts in there, right? Because they contain the chlorophyll. All right, so you can see all these green little bad boys in here. That's what makes the plant green. Okay. Cool. So if we zoom in on the chloroplast, we can see that it's clearly, pretty green and a little bit transparent. So first thing we need to notice is that it has 2 phospholipid bilayers. So that's not a phospholipid quad layer, which is the silly joke I always make. It's 2 phospholipid bilayers. All right, it has these round pancake-like structures that stack upon one another, and they're called your thylakoids. That's all right. All right, so an individual one of these is a thylakoid. All right, every single one of these are thylakoids. When we have a big stack like this, that's called a granum. All right. And then if you have multiple stacks that you're looking at, 1, 2, all right, something like that, they're your grana. All right. Cool. Other things to note are, of course, there's DNA in there, circular DNA and ribosomes. All right, because these can replicate independently. And lastly, the stroma, okay, which is sort of this intermembranous space. All right, in here, kind of like if we were looking at a cell and you're looking at the cytosol. All right, similar to that intermembranous space. All right, where there's nothing there. That is your stroma. The important things to remember here are, of course, the thylakoids and the stroma, because they're going to come back in the photosynthesis biochemical pathway. All right, we can also talk about factors that affect photosynthesis. Obviously, we're just looking at environmental temperature and pH, as well as the different things that plants need to survive. So, of course, carbon dioxide through gas exchange through the stomata. All right, water availability, pretty obvious. Plants need water to grow and survive. And, of course, light availability, so photosynthesis can occur. All righty, we should also mention that ADP and ATP are an interesting and important part of this biochemical pathway, and both biochemical pathways we're going to look at. So obviously we should know what ATP stands for, adenosine triphosphate. Okay, and that's when it has 3 phosphates. Triphosphate, 3 phosphates. All right. Whereas adenosine diphosphate here, obviously when you say tri, something has 3. If you say di, it has 2. All right. So in other words, that has 2 phosphates connected. So you can see that here, ADP and ATP on the top there. Okay. So the things we need to note here is that ADP can be turned into ATP, and that's through this enzyme ATP synthase. All right. So the enzyme is used to turn ADP into ATP. So it reattaches another phosphate, and it's PI here. You can see that. So it's called an inorganic phosphate. All right. So phosphate, inorganic, PI. And once that inorganic phosphate gets attached, you have your adenosine molecule, which is a bunch of different atoms together. So that's just bigger. And then you have your phosphate, 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 ATP. All right. So we have our process of C3 photosynthesis here. So we have 6 molecules of carbon dioxide, so CO2, 12 molecules of water, so H2O. All right, and there are inputs, our basic inputs. All right, we have the arrow meaning a chemical reaction is occurring, and then we have something above the arrow, which is light in this case, and it's going to be a biological catalyst there. All right. And of course chlorophyll, which is the thing that allows light to be absorbed. Note the colors in these 2 words. Chlorophyll is going to be green because that's what it is. And then the light is going to be any light that's not green light, because of course green light is reflected by anything that is green. Okay. Awesome. So our outputs here, super important one is going to be, of course, your basic sugars, such as C6H12O6, which is glucose. All right, we have 6 oxygen molecules leaving the plant as well. And then, of course, water molecules left over as well. Okay. And that's going to be either recycled by the plant or expelled via transpiration. Cool. And the thing to note is, as well about this equation, this is an unbalanced equation. So chemistry students, you would normally just be like, oh, what the hell? Cross this out, cross those out, and then you have 6H2O here. We do not need to know that. In fact, we want to learn it the way it is written here, okay? Because it allows you to remember the inputs and the outputs better. A lot of people will forget that water output by writing the balanced chemical equation. So we are learning the unbalanced chemical equation here. All right, we have some enzymes and coenzymes in photosynthesis. The main 2 you want to know are ATP synthase, as mentioned before. All right, and that helps create ATP from ADP. And then, of course, this one here, rubisco. All right. So what does that stand for? It stands for ribulose, bisphosphate, 1,6-carboxylase, oxygenase. All right. Silly long name, but the important part is the carboxylase. Carboxylase. Nice. And the oxygenase. It's very hard to write with the mouse, I'm sorry. All right. Cool. So it acts as both of those. All right. That's one thing to note. Carboxylase, oxygenase. That's the important part of rubisco that we're going to remember. Okay. And that works in the light independent stage of photosynthesis, also known as the Calvin cycle. But we're going to look at that in a minute. Alrighty. So let's talk about the stages of C3 photosynthesis. So firstly, we have the light dependent stage. As you can see, there's a little bit of light going in here. All right, and it's not an input, the light. It's a catalyst that's above the chemical reaction line. All right, so just keep that in mind. That is not an input, but it is required for the light dependent stage. All right, and this occurs in those circular green stacks that I mentioned before. Okay, so they're the thylakoids. We also have over here the light independent stage, which occurs in the stroma of the chloroplasts. All right, cool. So let's go through the inputs and outputs. So we're going to do inputs in red. We're going to do outputs in blue. Okay, so first of all, we're going to look at the inputs of the light dependent stage on the left here. So your first input, and remember, you don't really need to know the numbers for this, all right, but I guess it is there to help you as well. And you don't need to know this biological or biochemical pathway process. You just need to know the inputs, the outputs, and the location. That's all it says in the vcar.points and the vcar facts. All right. So firstly, we have ADP, so adenosine diphosphate, and inorganic phosphates. That goes in as an input. Okay, so that's input number 1. Pretty simple there. We have input number 2, which is this molecule, which is an electron carrier called NADP+. All right, that is also an input of the light-dependent stage of photosynthesis. All right, and the last input we have is water molecules, pretty simply. All right. So what is going to occur here? These inputs go in, obviously something occurs here, and then they're going to come out in a few different forms here. All right, something to note about the light-dependent stages, it uses the light to essentially catalyze the reaction of the splitting of water, also known as hydrolysis. Okay? So hydrolysis occurs, this water molecule gets, as you see, split. So the oxygens go one way, also known as in, and right out. And the hydrogens go another way. Boom. And they are going to attach here to NADP+. All right. As that occurs, we now have our outputs, which we're going to do, as I said, in blue. All right. So outputs of the light-dependent stage is going to be 18 ATP, all right, because we have our ADP and our inorganic phosphates coming together to make ATP. All right. Our second output here is NADPH. All right. So again, as you saw here, the hydrogens from the water molecules have now gone over here and have attached, as I said here before, to the NADP+. That makes NADPH. All right. And the last output, of course, is the oxygen, which has come from here, from the splitting of that water molecule. All right. I know this doesn't look too pretty, but hopefully you understand what we're going with here. So now we're going to look at the inputs of the light independent stage, also known as the Calvin cycle. All right. So we're going to go inputs in purple, Calvin. All right. Inputs, of course, 18 ATP. All right. Again, you don't need to know the numbers. And then NADPH are the inputs. They're your electron carriers. All right. Well, NADPH is the electron carrier. ATP is for energy. All right. And of course, the last one here, 6 carbon dioxide molecules. Specifically important are the carbons here. All right. So they're your inputs. What color? We'll go orange for our outputs of the Calvin cycle, also known as light independent stage of the of photosynthesis, which occurs in, of course, the stroma. All right, you have your 2 outputs here. So you have, firstly, glucose molecule. The thing to note here is that the carbons from here go in, and they go directly through, and they spin around and round and round, and then they go out to here. Alrighty, and that's the carbons within the glucose. And the other output, of course, is water molecules. All right, there's going to be some hydrogens and oxygens that need to be released. Alrighty, so the inputs, outputs, and locations of both the light-dependent and light-independent stage of photosynthesis. Okay, so we're going to continue on now, guys. On the next slide. Alrighty. So this image here, you don't need to know this, but this is a little bit more detail, and we're going to quickly go through it just to explain a few things. So what can we see here? We should be able to see that we have water molecules. We have our NADP, NADPH, ADP, ATP. We have ATP synthase over here, and a bunch of other things that we don't really need to know about. Okay? Firstly, water molecules are getting split. All right, so that's that process of hydrolysis. So boom, and you get your oxygen and hydrogen. All right. Meanwhile, electrons are essentially going through these things, and particular things are occurring. Okay, because really this is an electron transport chain through here. All right. So the point is, we get lots and lots of hydrogens coming over to this side. So H plus, lots and lots of hydrogens here. All right, lots and lots. So there's heaps of hydrogens on this side. All right, and what else can we note? This is a phospholipid bilayer. Okay, obviously this is in the chloroplast, in the stroma. So it should really say light independent stage, I've just realized. Anyway, so we have our phospholipid bilayer here. All right, so what's occurring? If we have lots and lots of hydrogen molecules over here, all right, and not that many hydrogen molecules over here, what's going to happen? There's going to be simple diffusion occurring. All right, so again, you guys haven't done this since year 11, but this simple diffusion is from an area where there's lots and lots of molecules, a high concentration, to an area where there's not many molecules, a low concentration. These molecules will go through, okay? So these hydrogen ions will go through ATP synthase as it's acting as a channel protein through the membrane here, okay? As it goes through, it spins the top of ATP synthase and it essentially shoots, well, in a way, it whacks the inorganic phosphates onto ADP and they get smacked together and then you get ATP. All right, one of your outputs. What else is occurring in the meanwhile? If you remember these electrons we spoke about over here, all right, they're coming through the two different electron transport chains and they're going kaboom. And they're bringing the hydrogens that are pulled across, all right, and they're attaching that with the hydrogen plus, the electron minus, and the NADP plus. All right, that's all coming along, and you get NADPH. You get ATP being created. All right. So yeah, that's really the deep processes of what's occurring. Do you need to know that? No. Does it help you understand the inputs and outputs and location? Yes. All right. So pretty important to remember that one. Cool. So again, light dependent stage inputs and outputs. Water molecules is an input. NADP plus is an input. ADP plus inorganic phosphate, which is one input, even though there's two different things. That's one input. All right, they're your inputs of the light dependent stage. The outputs of the light dependent stage are oxygen molecules, NADPH, and ATP. All right, again, you do not need to know the numbers. All right, let's look at the light independent stage, so the Calvin cycle here. All right. Again, Rubisco is here. We see that. There's a lot of things going on. And students often get misled or disenfranchised when they see something like this. You do not need to know this. All right, but what is important is certain things I'm going to point out. So we have our carbon here. So our 6 black molecules is going to be 6 carbon molecules. All right, also notable is Rubisco, that's in the middle here. All right, again, we're working on carbon, so that's acting as a carboxylase there. All right, it's not acting as an oxygenase at this point because there's a C3 plant in regular temperature. All right, the carbons spin their way through. Again, you don't really need to know what's going on, but they spin through and they get to this last point. And this last bit's really important too. That is your glucose. Remember the equation for glucose? It starts with C6. All right, 1, 2, 3, 4, 5, 6. Boom. And that is your glucose. That's the whole entire point of photosynthesis. Okay, if that does not occur, if there is no glucose made, photosynthesis is pointless or not even working. Okay, that's what the plant needs. It needs it to build. It needs it to use, for other sources of energy and things like that. It really needs that glucose. Okay, and again, Rubisco is also there. Very notable enzyme shown there. Alrighty, so to wrap up the video, we're going to look at the light independent stage inputs and outputs. So obviously an input is carbon dioxide molecules, NADPH, electron carrier, and ATP energy. What comes out, or the outputs, is glucose, water molecules, and I guess to an extent you can also think of these, but really it's just these two. But also NADP plus because the NADPH has been used, and ADP and inorganic phosphate because the energy from the ATPs have been used there. All right, cool. So really, you should be looking at this image here, guys, and you should be able to understand what's going on. Okay, so sunlight, biological catalyst, nice. All right, water, water's going in, oxygen's coming out. So this is obviously which part? The light-dependent stage happening in the thylakoid membranes. Beautiful. There's also NADP plus and hydrogen ions, as well as ADP coming from the carbon cycle, going back into the light-dependent stage. All right, then your outputs, ATP, NADPH, all right, and of course oxygen, which is leaving the plant, goes into the light-independent stage, the Calvin cycle here. You have carbon dioxide entering the plant as well through gas exchange. Beautiful. So they're your inputs, here, here, and here. That spins around and, of course, creates the outputs, which would be water, all right, H2O, NADP, and of course the hydrogen, and ADP, and inorganic phosphate, and of course the most important and the star of the show, glucose. The whole point of photosynthesis. All right, so you should be able to go through this picture. Pause it if you have to. Have a good look at the different labels, and understand it. All right, I know this one's been a long one, guys, but you can watch this multiple times. You can slow it down. You can speed it up. Please, if you have any questions, ask me. Yeah, good luck. Have a nice day. Like, comment, subscribe.